Okay, so this is the final lecture of our semester, and congratulations if you made it this far, listening to my monotone voice over all these lectures. But today we're going to be talking about translation, and I think the book calls this protein metabolism, but a better name for it is really translation. It's how we get the information that was transcribed from DNA to RNA, how we get that from RNA into an amino acid chain that becomes a protein. So if we look back to our RNA polymerase and the messenger RNA that we're making, what that messenger RNA does is its job is to go from the nucleus where it was made, and then go to the ribosomes in the cytoplasm and be translated into protein sequence. Okay, and we'll talk about the exact details of this process during this lecture. First, I've included a few slides here to remind ourselves what amino acids are. Okay, they are molecules that have a carboxylic acid and an amino group attached to an alpha carbon, which then is also attached to something, an R group, a side chain, and there are, in this case, a methyl group. It would be alanine. Most natural occurring amino acids are the L version. Okay, so we don't really have to think too much about stereochemistry with them. Okay. And the ones that are involved in protein synthesis, we call these the common amino acids, or the ones that are involved in proteins. There are 20 of them. And their symbols and names are given here, so just a refresher for that if you need it to go through that, because these will be important now again. There are different groups of amino acids depending on their side chains. So the ones that have nonpolar side chains are shown here. They're glycine, alanine, proline, valine, leucine, isoleucine, and methionine. The aromatic side chains are phenylalanine, tyrosine, and tryptophan. The polar uncharged side chains are serine, threonine, cysteine, asparagine, and glutamine. Then we have positively charged side chains of lysine, arginine, and histidine. And then finally, the negatively charged side chains of aspartic acid and glutamic acid. Okay. There are many different levels of protein structure, but the most basic is the primary structure, and that's the sequence of amino acids. So amino acids coming together and bonding together in a chain via peptide bonds, where you have the carboxylic acid of one amino acid attaching to the amino group of an adjacent amino acid. Okay, so when that happens, you get what are known as peptide bonds. Okay. And during the process of translation, this is what's occurring. You are sticking amino acids together and forming these peptide bonds. So these we'll see these again when we look in the details of translation. Okay. Translation, what translation is really an mRNA, messenger RNA guided protein synthesis. It's very complex. Okay. It's said that there are about 70 different ribosomal proteins, and that's not counting those involved in amino acid activation or other factors that do initiation, elongation, or termination. Okay. Overall, it's estimated that there's about 300 different macromolecules involved in this process, so it's very complex. And because of that complexity, it uses a great deal of the cell's energy. So it's estimated that it's about 90% of the cell's energy is going to this protein synthesis. Okay. Even though it is complex, it happens amazingly fast. In E. coli, It's estimated that a 100 amino acid protein can be synthesized in about 5 seconds. So there were 3 early advances in understanding protein synthesis. One, it was determined that proteins were synthesized at ribosomes. 2, amino acids, it was found that the amino acids were activated for synthesis by attachment to tRNA or transfer RNA. Okay. And they're done, this is done by proteins called amino-acyl tRNA synthetases. So amino acids aren't just freely floating around like nucleotides to be put into their protein chains. They actually have to be attached to transfer RNAs. And the transfer RNAs act as an adapter to be able to translate messenger RNA code into an amino acid. And Crick of the Francis Watson and Crick team was the first to sort of guess the existence of transfer RNAs. So in the early 1950s, he kind of guessed that protein sequence was encoded by nucleotide sequence. The molecular nature of the players of this remained unknown at the time. In 1954, he proposed that there's an adapter that can bond to the messenger RNA and then carry an amino acid. Okay. So he really did do a good job of hypothesizing transfer RNAs. Well, how a messenger RNA is read is that you're read in specific what are called codons. Okay, and codons are 3 nucleotides long, so 3 bases, in this case of RNA, since we're talking about messenger RNA, would make up a codon. Okay, you need to encode for 20 amino acids. So if you just do the numbers, if you tried to do 2 amino acid or 2 nucleotides, it would not be enough. So you could only make a possible 16 combinations with that. 3 nucleotides, you can make 64. So that's plenty to be able to code for 20 amino acids. So that's one of the ways that they sort of guessed that it was 3 nucleotides just by doing the math. But as we've found out since then, it is 3 nucleotides per codon. And it's read in a non-overlapping fashion. So that's a pretty important thing to note as well. So if we had a string of messenger RNA, the codons would be 1, 2, and 3, and not 1, 2, and 3. So they do not overlap. You can imagine if they did overlap, the complexity of that would be quite a bit higher, and you'd sort of be, depending on what amino acid you have first, it would kind of limit your choices for the next amino acid, which would not necessarily be good for protein synthesis. Okay. So a messenger RNA is read from the 5' to 3' region, okay, when it's translated. If we have an insert, let's say we have an insert of 1 nucleotide. So in this code up top, this is our original code. If we insert 1 more nucleic acid there, we change the reading frame. Okay, and so that changes this codon from a UAC to a UCA, right? Which makes a big difference to not only this amino acid, but it changes every subsequent amino acid codon in after that has been added. Okay, the same thing with the deletion. If you take 1 nucleotide out of that sequence, it's going to change the codon for every codon after that. Okay. If you have an insertion and a deletion on that odd chance, you then could change 2 codons, but the rest of the reading frame would be restored. So that's something to keep in mind when you're thinking about how do mutations in DNA, how do they change what a protein, the protein product is. Right, so there are 3 reading frame possibilities. You can start from, if you start from the first nucleotide in this messenger RNA sequence, right, our codons would be given here. But what if you started from the second nucleotide and then did those 3 as your first codon? It would change what your codons are. Okay, so that's what's referred to as a reading frame, where you start reading the codons. Right? And really, to get around this problem, everything has to be defined by a start codon. Right? And the reading frame, really typically, it doesn't change during translation. You start at the start codon, and then the reading frame continues on from there. Right, so what is the start codon? Well, the start codon is, it is AUG, and it starts with the amino acid methionine. So all proteins start with that. And that, again, is to make it consistent, a consistent way to start translation. Okay. All amino acids, except for methionine and tryptophan, interestingly enough, have more than one codon, right? We said that with codon of 3 nucleotides in length, you can have 64 codons, right? That gives some ability for amino acids to have more than one codon, and they all do except for methionine, and that's because it's is the start codon, and tryptophan have more than one. There are 3 stop codons, and those all are start with U. So UAA, UAG, and UGA are stop codons. All life, for the most part, in this class you can just consider this all life on this planet, uses the same genetic code, the same codons, which is kind of interesting. And that really leads to the fact that life supports the theory that life developed one time on the planet. So a random sequence would have a stop codon approximately every 20 frames. So if there are 3 of them, 3 stop codons, 64 possible codons, if you're just random about it, about every 20 frames, you're going to have a stop codon, okay. Which would not necessarily be a good thing. All right. If you observe an ATG, or the start codon, and no stop codon within 50 codons, then that's a potential open reading frame, or ORF. Okay. And ORFs, it's just a fancy way of saying that is a gene or a piece of DNA that encodes for a protein. Okay, so if you ever see ORF, don't be thrown off. It's just a fancy way of saying a sequence that's encoding for a protein. So if we are given a simple messenger RNA here, what amino acid sequence does the following messenger RNA encode? So if you're given this type of a problem, first you have to find a start codon. And remember that start codon would be ATG in DNA, but in messenger RNA it's AUG. Okay, so we scan for our first AUG, and that would be here. Okay, so then our open reading frame would start after that. So we would start with AUG, which is methionine, then it would be UUC. So we go to our U's. So UUC would be phenylalanine, and GGU, I'll go to the G's, GGU would be glycine. And when we're doing this, we're translating that code. We're starting 5' to 3' on the messenger RNA. We're building an amino acid sequence from the N terminal to the C terminal when we do this. So translation is creating a protein chain from the N terminal to the C terminal. This genetic code is universal. It's used by prokaryotes and eukaryotes across species. Mitochondria, contain DNA. They use slightly different code. And one of the differences is that UGA encodes tryptophan in vertebrate mitochondrial DNA instead of a stop. So in that case, you have tryptophan has 2 codons in mitochondrial DNA. And AGA or AGG encodes stop in vertebrate mitochondrial DNA instead of an arginine. So those are just 2 differences. You won't need to know those specifically in this class. Okay. Mitochondria also encode their own transfer RNAs, and they use 22 of them instead of 32 as in the cytoplasm. So here are some known variant codon assignments in mitochondria. Again, not important to know these in this class. It's just, kind of interesting. It's one place you do see some variation in codon assignments. Okay, how was the genetic code cracked? Well, these people, this group, used synthetic sequences of messenger RNA used with E. coli cell extracts to create polypeptides. Okay. And they observed the frequency of incorporation of certain amino acids shown here. Okay. Lysine was 100. And then they tentatively assigned nucleotide composition for the corresponding codon. And they kind of were able to piece together what the codons looked like approximately. Okay. They knew that lysine, because it gave a frequency of 100, and the same frequency in their messenger RNA for AAA, that sequence, they could assign lysine to that. Right, so they also determined certain trinucleotides that induce specific binding of amino acyl tRNAs to ribosomes using C14 labeled amino acyl tRNA. And they found that UUU was the phenylalanine, AAA again was lysine, CCC was proline. So they used these 3 nucleotide repeats and found which amino acids they were. Most amino acids have more than one codon, as we've said before, and only methionine and tryptophan are the ones with single codons. Some codons are less subject to causing mutations in an amino acid sequence because there's some degeneracy or similarity in the abundance of the tRNAs. And we'll kind of go through that when we see the actual table again. And if you're curious of the number of codons each amino acid has, that's listed here. So things like serine, leucine, and arginine have the greatest degeneracy. They have 6 codons for each of them. But most of the amino acids have, like between 2 and 4 codons. So the codon sequence is complementary with the anticodon sequence. So the anticodon sequence is the sequence that is contained within the transfer RNA. Okay, so the codon is in the messenger RNA, and it's going to base pair with the anticodon, which is in the transfer RNA. And it does that via those hydrogen bonding interactions that we've seen before in Watson-Crick base pairing in DNA. The alignment of the two RNA segments is again, anti-parallel. Right, one slight difference you have with the interaction between tRNA and mRNA is what's known as wobble, and that occurs at the third base in the codon. So the third base of a codon can form base pairs with a complement in tRNA that are not considered the normal sort of Watson-Crick base pairs. Okay. Some transfer RNAs contain a special base called inositate, which is labeled I, and that can hydrogen bond with U, C, and A. Okay, so it can hydrogen bond with all three of these. These H bonds are weaker and were named wobble base pairs by Crick because they're not held as tightly. Okay, so example, in yeast, CGA, CGU, and CGC all bind to the transfer RNA for arginine, which has the anticodon GCI. So that I in the wobble base position can interact with A, U, and C. Okay. Sequences are usually written 5' to 3', but the anticodons here are written 3' to 5' just to illustrate how they match up with those messenger RNA codes. So what does a tRNA look like? Basically, we can draw a picture of it like this, and it makes sort of a T shape, cruciform type shape, which I like to think of as a T because it's transfer RNA, tRNA. Okay, and down here at this little loop by this hairpin at the base is where the anticodon is. Okay, and so we have that position 3, 2, 1 in the anticodon, and again that's reading 3' to 5'. We have our anticodon, and that matches up with a 5' to 3' codon. So that wobble base, in this case, if we have the first two base pairs of our codon, we can have A, G, or U and C. So one reason inositine, which can form H bonds with U, C, and A. So if we look at, for instance, things like isoleucine here, AUC is the codon there that for isoleucine, but it can also be AUU or AUA because of that wobble position. Right, so the specific base at in the wobble position of an anticodon will determine the number of codons that tRNA can recognize. If the anticodon has a C there, it recognizes one or an A. 2 codons would be recognized if you have a U. It can bind with A or G. If you have a G in that position, also 2, it can bind with C or U of the codon. And then 3 codons, if there's that special base I there, it can bind AU or C. Right? So that's why you see the differences in the number of codons that can be recognized. But the genetic code is resistant to mutation. So these degenerate codes allow certain mutations to happen, but still code for the same amino acid. And these are termed silent mutations, or also known as polymorphisms, single nucleotide polymorphisms, where you get a different nucleotide in the DNA, but you still get the same amino acid in that protein. Okay, a mutation in the first base of a codon usually produces a conservative substitution. So if you have a G going to an A, that's amino acid change in that first position. You change from a valine to a leucine, but they're the same type of amino acid. Right, so if we look at the genetic code, for instance, if we just use that example of isoleucine again, right, if you have a change in that third base pair of isoleucine, you're likely to continue to be an isoleucine. Some other examples, if you pick out some of these, you can actually change the middle base and end up with the same amino acid as well, although I did not pick any of those out ahead of time. Something like an arginine is a good example how you can change the first base. If you change an A to a C in these two, you're still going to get arginine. Okay, so there is some degeneracy to the code, which leads to resistance against mutations. Another way of looking at this picture, our transfer RNA, our codon, anticodon, written red position 3, 2, 1. That's because you're going 3' to 5' backwards. And then your messenger RNA codon is read 1, 2, 3. And that wobble position there is the last one. And probably more importantly, the table telling you if you have a wobble base, the base in the wobble position of the tRNA, which bases in the mRNA can it recognize? So the strength of these H bonds are variable, and they depend on the orientation, the distance, and the effective charge or electronegativity. Okay. So depending on this, it's one of the reasons why this wobble position, you can have other base pairing that we don't normally see. Okay. The first two bases of messenger RNA code from strong Watson-Crick base pairing with the tRNA anticodon. The first base of the anticodon, the one that's on the 5' position, determines the number of possible recognized codons. Okay, if the first is a C or an A, that's specific base pairing, only one codon recognized. If it's a U or a G, that's less specific, so 2 codons can be read. And if the first is an I, that's even less specific, so 3 codons can be read. Right? So, and when we say first base of an anticodon, that means the wobble position. So that would be the last base in the codon. Okay, if more than one codon encodes the same amino acid and different bases in position 1 or 2 of the codon, then they use different transfer RNA. Okay. And because of this, there are 32 transfer RNAs are needed to translate 61 codons. Right? There's 61 codons because 3 of them are stop codons. Okay. Weaker hydrogen bonding at this wobble position helps with the speed of translation. So that's thought to be why it the benefit of having that wobble position. Okay, so what anticodon sequences bind to the codon below? So AUG, GGA. Right, so if we use AUG, that's our start codon, it can bind 3' to 5' with the anticodons UAU or UAC. Right. However, UAU would also recognize AUA. Therefore, the tRNA for methionine has UAC. right? So that is looking at the transfer anticodons that you have in a methionine. Okay. And so reading 3' to 5', you'd want this transfer to be a UAC, so there's only one possibility for methionine. If you look at the frequency, this is in E. coli, but you can look at the frequency of how the codons are used within the genome of an organism. So this is E. coli, right? And things like methionine that has one codon, right? The frequency is just going to be 1. It always uses that when there's a methionine. But if we look at, let's say, isoleucine, that's been our example so far. There are 3 possibilities, right? And the codons, they're not equal in their usage, right? They're this first one, AUU, is used the majority of the time, 58% of the time. The second one is used 35% of the time, and then this one, AUA, is used only 7% of the time. So when you're designing plasmids to express a protein in a bacteria cell, and the protein is let's say the protein's a human protein, sometimes you have to take into account the differences in the codons used inside an E. coli cell. So you have it optimized, the coding optimized for E. coli. For instance, if there was a lot of these AUA sequences in your protein, maybe humans use that more often, you'd want to change that to one of these other 2 codons. And then likewise with the stop codon, the bacteria cells really recognize that UAA as their stop, as opposed to UAG, which it never uses naturally. Right. Some messenger RNAs are edited before protein synthesis, and that involves alteration, addition, or deletion of nucleotides in the messenger RNA. We talked a little bit about cutting out introns as well at last lecture. Okay, editing. These are different types of edits, though. Editing uses what are termed as guide RNAs or gRNAs that temporarily bind to the messenger RNA and act as a template for the editing. So here's a way that we can make this process more complicated than it already is. So if you have an editing here, this is our messenger RNA, and our guide RNA would bind to that and hybridize, and then at certain points it's going to change and insert different nucleotides in that messenger. Okay, not something that we're really going to focus on in this class, though. What this processing can be used for, though, is to encode more proteins for a given gene. So something like that viral genome that we talked about last lecture, the GAG reading frame is shown here, but the polymerase reading frame is shown here. So just being able to edit this in using the same DNA, or in this case it's a retrovirus, so the same RNA genome can encode for multiple protein products, and that's one of the ways. A protein synthesis video is included here, so if you want to see this visually, this is a good video I found that shows this process. So moving on to the second section of chapter 27, these are the 5 stages of protein synthesis. Okay, so we got a general overview of it, and now we're going to look in more detail how this occurs, how translation occurs. Okay, so first you need the activation of amino acids, and that they have to be added to transfer RNAs to charge the transfer RNAs. Then you have initiation of translation. That's where the messenger RNA binds to the ribosome, and the AUG start codon will start the process. Next, you have elongation, where you're creating making your protein chain longer by one amino acid at a time, followed by termination and release. So that occurs when the stop codon is encountered. When that happens, the protein is released from the complex. And then finally, you get folding and post-translational processing of that protein. So you can have certain amino acids removed or cut off. Others have modifications added to them, and then the overall fold of the protein's allowed to fold into its active native form. Right. Seeing that visually is probably a little bit better, and so this figure 2713 in the book shows that. So the activation of amino acids, the tRNAs are what's referred to as amino acylated. Okay, so they have these amino acids attached to them at their ends of them. Okay. Right. The initiation starts when messenger RNA, shown here as this green thread, binds to the small ribosomal subunit. So there's a small subunit and a large subunit that we'll be talking about in this class. There are more subunits involved as well, but these are the ones we're primarily focused on. So the small subunit will bind that messenger RNA at the start codon and the start tRNA. Okay. Right. Then when that happens, the large subunit can then bind, and then we get initiation. Okay. Elongation happens in successive cycles where you have a charged tRNA binding, and the peptide bond then forms between, in this case, the first amino acid, which would be methionine, and the next amino acid on the adjacent tRNA. So these are coming in, binding, and then the ribosome's catalyzing the formation of peptide bonds between those amino acids. Okay. Right. Termination occurs once elongation goes on until you reach this stop codon. So you reach a stop codon, and then we get termination. So the messenger RNA and the protein dissociate, and the ribosomal subunits sort of open to allow that to happen. Okay, those are recycled, those are used again, the ribosomes, and the protein then can fold into its native state. And it's thought that the proteins really do kind of stay unfolded when this is occurring, because if they immediately fold up, that could, make the translation process a little bit more difficult. So there are certain chaperone type proteins that keep these proteins unfolded until they are fully synthesized. Okay. These are the components required for these 5 major stages of synthesis in E. coli. So the 5 stages of translation in E. coli. Right. Stage 1, that activation of amino acids, you need the 20 amino acids. Then you also need 20 amino acyl tRNA synthetases, so enzyme that will take each of the amino acids and stick them to tRNAs. You need 32 tRNAs, you need ATP and magnesium. Okay. All right. So you can kind of look through this list to see exactly what you need at each stage. It's kind of a good overview. As we go through some of these in more detail, if you look back at this and make sure you kind of understand what piece each of these plays in each of these stages, it's a good review. Okay. So the ribosome is a key player in protein synthesis. It makes up an estimated 25% of the dry weight of a bacteria cell are ribosomes. And they are made up of approximately 2/3 ribosomal RNA and another about 1/3 of protein. So it's a mixture of protein and RNA that come together to form these. The ribosomal RNA forms the core, and it also does the catalysis of the peptide bond formation. Ribosomes are made up of two subunits bound together. In bacteria, they're called the 30S and the 50S. Okay, so the one's smaller and one's larger. Okay, the 30S and 50S, what those terms are, they are really from when these molecules were discovered. And they were discovered and separated out using analytical centrifugation, and the layers which they were sort of concentrating in this centrifugation were termed 30S and 50S. That kind of gave the relative size of each. So that's where those numbers come from. So just keep that in mind. In eukaryotic cells, those numbers are different. Okay. If you're curious about these individual subunits, 30S and 50S, here are the number of different proteins involved in them, and the protein designations, not that you'll ever need to know that, at least in this class, and the number of and type of ribosomal RNAs. So in the 30S subunit, you get the 16S ribosomal RNA. In the 50S subunit, you get the 5S and 23S ribosomal RNAs. So it gets more confusing still, right? Because in that subunit, really what you care about in terms of translation is the rRNA. That's, the core of it. And in that 30S subunit, it has a 16S rRNA. In the 50S, the larger subunit, has two pieces of rRNA, a 5S and a 23S. Okay, and that's specific for E. coli. This is a picture that illustrates the differences between the bacteria and yeast ribosome. So here's the bacteria. We have the 50S subunit and the 30S subunit, and the rRNA is shown in this sort of filled in model, and the protein is kind of in this wire model. Okay, so the 30S subunit and the 50S subunit and the 30S subunit are shown here. In yeast cells, that's called the 60S subunit and the 40S subunit. So they're a little bit bigger subunits, but again, there's a larger one and a smaller one. Okay. Well, the tRNAs bind in three different sites, and the sites are E, P, and A. Okay. We'll see other figures that kind of explain these, but this is just using a crystal structure to show the relative positions of those. And the ribosome is a ribozyme. It's a RNA, a piece of RNA, in this case RNA and protein, that catalyzes a reaction. And the RNA itself is what's interacting. The messenger RNA is what's interacting with the transfer RNA. And the messenger RNA kind of fits in this groove in the ribosome. This is just another picture showing a ribosome and a transfer RNA inside of it, and this is where that transfer RNA is removed. You can see that cleft there, right, where the messenger RNA kind of goes in there, shown here, and then the transfer RNA can come in and fit and interact with the messenger RNA. Okay, so these rRNAs have complex secondary structure. So 3 single-stranded ribosomal RNAs in E. coli have specific 3-dimensional structures with extensive interchain base pairs. And the shape of these ribosomal RNAs are highly conserved. Okay, and so how they look in bacteria compared to archaea, compared to eukaryotes, you can see that these patterns they follow look very similar. Right? Eukaryotes, they're a little bit longer, so you get longer pieces, like for instance here, and then maybe this loop's maybe a little bit longer, and this red piece might be a little bit longer, but the overall shape of them is very similar, the overall secondary structure. So if we're comparing ribosomes in eukaryotes and prokaryotes, overall they're very similar. They still have two subunits in eukaryotes, with the mRNA running in between the two. They're larger, they're more complex, and they contain a larger number of proteins. Chloroplasts and mitochondria have the ribosomes that are simpler, and in fact they're actually simpler than those that we see in bacteria. And the thought there is that chloroplasts and mitochondria are, evolutionarily very ancient kind of bacteria cells that were taken up by other cells. So that could be one reason why their ribosomes are simpler than those in present-day bacteria. If we look at the bacterial ribosome, we give that term is referred to as 70S for the complex. Eukaryotic ribosome is 80S. Again, that's kind of a measure of the size of it. The molecular weight is shown here on each of them, and you can see it is bigger in the case of eukaryotic cells. The subunits individually in bacteria, that was the 50S and the 30S. In eukaryotic cells, it's the 60S and 40S. So they just added 10S to each of those. Okay. And the actual pieces that make up these, the rRNA pieces that make up the subunits are important. So in bacteria, that was to make up the larger 50S subunit, you had the 5S rRNA and the 23S rRNA. In eukaryotic cells, you have a similar 5S rRNA. You have a 28S rRNA, and then you also have a third piece, 5.8S rRNA. So there are actually three pieces of RNA in the large subunit in eukaryotes. In the smaller subunit in bacteria, it was the 16S rRNA. In eukaryotic cells, it's the 18S rRNA. Okay, so those are the makeup of ribosomes. And now let's talk about transfer RNA, the tRNAs. Okay, so single-stranded RNA of about 73 to 93 nucleotides in both bacteria and in eukaryotes. So these transfer RNAs are single-stranded, and they're made up of between 73 and 93 nucleotides. They have that clover leaf or cruciform or T-shape to them in 2 dimensions. And in 3 dimensions, it makes what's referred to as kind of a twisted L shape. Most have G, the nucleotide G, at the 5' end. And all of them have CCA at the 3' end. Okay. And one thing about the tRNAs is that you get modified bases as well, especially methylated bases. Okay. There are different parts to them. One, if we think of where the amino acid gets stuck on to the transfer RNA, that's referred to as the amino acid arm. And that's because it has the amino acid stuck there or esterified via that carboxyl group on the amino acid to the 2' or 3' OH group of the A in the terminal CAA codon. Okay, so on that last A in that final CAA codon is where the amino acid is stuck. You have an anticodon arm that contains your anticodon in that two-dimensional picture that's at the bottom. There's two more pieces, a D arm and a T psi C arm. Okay, the D arm is called that because it contains dihydrouridine, which is a sort of modified base, which is abbreviated as D. This contributes to folding. The T psi C arm contains pseudouridine, another modified or special base, which is abbreviated with the Greek letter psi. And that's bonding between the base and ribose. Okay. And it helps also in folding. So really, these probably aren't as important. The most important arms to know are the amino acid arm and the anticodon arm, because those are the ones directly involved in translation. Right, so if we look at this in a picture, a two-dimensional picture, right, our amino acid arm is up here at the top. It's at the 3' end of this transfer RNA, and we have that anticodon there at the end, which I believe is CAA, or at least it was in the last picture, the last slide. Okay. And that final 3' A is where your amino acid gets stuck to. Okay. Then down here is the anticodon arm, and again your anticodon, if we look at it in the 3' to 5' direction, right, because that's usually the way we're going to look at these, because they're matching 5' to 3' on the codons, and the wobble position would be there. Okay. The other arms, the T psi C arm and the D arm, are shown here, and again you have these bases, these special bases, dihydrouridine, which has the extra hydrogens added to it, a double bond removed from uridine, and then the T psi C has this pseudouridine residue added to it, where you have this particular base located there. In pink, what's listed in pink are conserved nucleotides. So those are thought to be important. These down here primarily in structure of the tRNA, and then up here it's important for attaching the amino acid. Okay. So how are these processed? Well, there's at least one kind of tRNA for each amino acid. Some tRNAs recognize more than one codon, and again that's because you have that wobble position. So in this case, what codons would this tRNA recognize? Well, remember, I, at the wobble position, can recognize 3 different nucleotides. So then our codon that we're going to be recognizing for this one would be GCA or GCU or GCC. Okay. And G is the only nucleotide that the I cannot recognize or bind to. Okay, all of these tRNAs are bound to alanine. Right. Three-dimensionally, this is what the structure looks like. So this is where that twisted L comes into play. Probably not super important for our class, but just seeing the positions of where the amino acid and the anticodon are relative to this is probably all you need to know from this. A lot easier to look at in the two-dimensional form, though. So let's go through the steps of translation in some detail. So the first step here is activation. This is where the tRNAs are charged. So each amino acid has its own enzyme called an amino acyl tRNA synthetase, which loads it onto tRNAs. Okay, there's 2 classes of these based on the structure and mechanism. Not important that you know which amino acid is in which class, but the table is given here. Okay. You get the same product from both classes of these amino-acyl tRNA synthetases. Okay, so it doesn't matter which class your amino acid uses, which class of enzyme, the end product's going to be the same. And again, that end product is attached to that last 3' A nucleotide in the tRNA. So all of this sequence is really sort of irrelevant. What we really care about in the tRNA is the anticodon and the A group at the very 3' end of it. Okay, and that A group at the 3' end will have a phosphate group. In particular, I think it's a triphosphate group. And you'll be attached to an adenine nucleotide. Okay, excuse me, here it is. The adenine nucleotide with that triple phosphate group is then what's used to transfer your amino acyl group to it. So this is our amino acid, and now it's esterified to that, in this case, 3' hydroxyl on our final base of our tRNA. Right? And it can be the 3' or sometimes it is the 2' position. Right? So the overall general mechanism here is you have your adenine ATP that you sort of activate your amino acid with. So you have your carboxyl group of the amino acid do nucleophilic attack on the alpha phosphate of your ATP. Okay, so you're using ATP. Then you get an amino acid attached to an AMP, so amino acyl AMP. Right? And then depending on the class of enzyme that you use, depends on what position you're attaching it to. So class 1, you get a 2' a nucleophilic attack from the 2' hydroxyl on the final adenine base of your tRNA. So that attacks your terminal carboxyl in your amino acid, and then you get the release of AMP. Class 2, the only difference is you're starting with the 3' hydroxyl. Okay. But again, knowing the differences between those two, not super important. Just know it's the same basic mechanism. It's just 3' or 2'. But in our class, doesn't really make a difference. Okay. Right. In both cases, you get release of AMP, and you have amino acid stuck to the final terminal adenine base in your tRNA. In class 1, the amino acid can stay at the 2' position, or you can get what's termed trans-esterification, where the 3' hydroxyl then attacks it, and it switches from the 2' to the 3' hydroxyl, but again, not super important in our class. So our final ready charged tRNA ready for translation is shown here, where we have our tRNA attached via this ester bond to the 3' position of our terminal adenine base at the 3' end. Okay, so this is then the active site, which the NH3 is the amino group, which then can be open for the peptide bond formation with the next amino acid's carboxyl group. Okay, proofreading does happen in translation, and it happens when you're charging the transfer RNA. Okay, one of the places it happens. Okay, so this doesn't happen in the ribosome. It happens in the actual charging. So in the case of isoleucine tRNA synthetase, there's 3 checks. So the binding site checks for interactions, and the reason it's doing this, right, is because isoleucine is pretty similar to like the amino acid valine, for instance. There's just one extra methyl group on the side chain. Okay, so those are being pretty similar. Valine can fit into the active site that an isoleucine can fit into. So there are 3 ways to check this that the enzyme does: binding site interactions. There's a proofreading of this enzyme where the site fits the valine AMP but not isoleucine AMP. So if valine does get in there, the valine AMP will fit in the proofreading site, and then the incorrect amino acid, it'll term it as incorrect because it fits in there and it'll be hydrolyzed. Okay. This is often, these tRNA synthetases are often referred to as having a second genetic code because they have to be specific for both the amino acid and the tRNA. So matching each amino acid with the correct tRNA can be viewed as a second form of a genetic code. Okay, the code is in the molecular recognition of a specific tRNA molecule by a specific synthetase. Only a few nucleotides in that tRNA confer the binding specificity. Okay, those are the anticodon region, and so the primary determinant in the alanine tRNA is a single GU amino acid in the amino acid arm. So this position here is the primary determinant in the alanine tRNA molecule. So that's, pretty remarkable that something so simple as that one base pair can give specificity to which amino acid gets put on it. Right, so again, just another picture of this in the specific sort of modified bases that are included. What's really important, though, again, are the anticodon and the amino acid arm at the 3' end. There are additional genetically encoded amino acids. So 20 that are encoded are common in all organisms and wind up in protein synthesis. There are 2 additional amino acids that are found in a few proteins that are genetically encoded. One is selenocysteine, and this is formed after charging with a UGA stop, recognizing tRNA with serine in both bacteria and eukaryotes. And there's this pyrrolysine, which is directly attached to a tRNA that recognizes that UAG stop codon in some archaea. So just not important in our class, don't have to remember these, but just to kind of like point these out to be inclusive. Right, and the structure of those are shown here. If you do see examples of selenocysteine if you do go on to like protein biochemistry and study proteins in eukaryotes. Relatively rare, but you do see those. Right. And this is just a figure in your book showing how some of these rarer amino acids kind of come to be charged. Okay, stage 2 is the initiation of translation. So you have 2 different tRNAs for the AUG start codon. The tRNA methionine is only used for the start codon, and that is this formylated F-methionine. That's the one that's used for the start codon only. It's special. And then you have another tRNA for methionine when the methionine residue is an internal one within the protein. Okay, they have the same amino acyl tRNA synthetase loads both of them. Then you have another enzyme that adds the formal group. So it's a transformylase that adds that formal group to the tRNA after the addition of methionine. Right? So not in eukaryotes except in mitochondria and chloroplasts. So this special formal methionine is really, in our cells, it's really in the mitochondria where it's important. And it's termed N-formal methionine. Okay. So initiation happens in three steps. And this is specific, this figure is specific to bacteria. So we're starting with the 30S subunit, the small subunit. Okay. And what's required here, you need the 30S subunit, ribosomal subunit, you need the messenger RNA, you need initiation with a FMET tRNA, you need initiation factors 1, 2, and 3, so IF1, IF2, IF3. You need GTP, and GTP is used for energy, much as an ATP would be. And then you need the large 50S subunit, and you need magnesium. Right, so how do all of these occur? Well, let's look at the figure first. In the 30S subunit, you have the P site and the A site. Right? The IF1 and IF3 will bind to your 30S subunit. That allows your messenger RNA to bind, and it loads it up so that the start codon, the AUG, is in what's termed the P site. Okay, when that happens, IF2 with GTP bound can bring in the FMET tRNA, right? And that FMET tRNA loads up to the start codon in your messenger RNA. Right? The 50S subunit then comes in, and you get that GTP cleavage, and you get the release of all those initiation factors, and then you have what would be similar in transcription to a closed complex with both ribosomal subunits now sandwiching the messenger RNA, and your first FMET tRNA in the start codon in the P site of your ribosome. Okay, so the first three steps of initiation, just in words, they're written here. I basically went through these, so you can kind of just have this to look through for review. One thing I didn't mention is the initiation AUG codon in the messenger RNA is guided to the correct position by what's known as the Shine-Dalgarno sequence. It's the region in the messenger RNA that's complementary to the sequence in the ribosomal RNA. So that's what allows it to line up correctly. In some examples of what those Shine-Dalgarno sequences are shown here, and they're rather close to the start codon. Right, what the IF3 subunit does is it keeps the 50S or the IF3 initiation factor 3. It's keeping the 50S subunit from binding prematurely. So that's its role. And the three sites, we've mentioned them before. You have an A, P, and an E site. What they stand for, and we'll see this in some more detail when we talk about elongation, but what they stand for is A is amino acyl, P is peptide, and E is exit. Okay, so step 2 of initiation, you have that formal methionine tRNA binding in the P site with help from IF2, and then step 3 is when the large 50S subunit combines with the 30S subunit forming the initiation complex. And to do that, IF2 hydrolyzes GTP. Right? And again, shown here in some more detail. Right? FMET does not fit in the A site, so it not incorporated into the middle of a protein. Okay, so it has to fit in that P site, and that's one of the reasons why it's only used at the very start. And IF1, its job is to block that A site, so no other tRNAs bind there before you can get this initiation complex formed. Okay, so that's in prokaryotes, and in eukaryotes, it's a little bit more complicated because there's more initiation factors. It has a step that actually circularizes the messenger RNA, so it's not just hanging out as a thread, but it becomes circular. And this is outlined in figure 2727. But for this class, we'll just focus on the process in prokaryotes and understand that in eukaryotes, it's similar but more complex. Right, and here's kind of that figure there. Right, and thankfully, we don't have to know all of these because we get a lot more abbreviations, a lot more of these other cofactors involved. Once we get an initiation complex, you see that it looks very similar to what we've seen in the bacteria, where you have two subunits, messenger RNA sandwiched between them, a start codon matching up with our first methionine. And also you have E, P, and A sites. Okay, the third stage of translation is elongation, and this again is in 3 steps. Okay, you bind the second tRNA, you form a peptide bond between them, and then you get translocation. And what you need for elongation, you need the initiation complex to have formed. You need aminoacyl tRNAs as your substrate. You need what are termed elongation factors, and you also need more GTP for energy. Okay, so the first step in elongation, aminoacyl tRNA is going to bind to the complex of elongation factor 2, or TU, so that's EF standing for elongation factor, and 2, not the number, but T and U. Okay, this also carries a GTP. Okay, then the aminoacyl EF2 GTP complex binds to the aminoacyl A site in that initiation complex, so in the ribosome. Okay, after GTP is hydrolyzed, that EF2 GDP leaves the ribosome. So if we look at this, our EF2 GDP complex with our aminoacyl tRNA comes into the A site. Right? The initiation complex always has that FMET tRNA in the P site. So our next one is coming in adjacent to that. So this binds that complex. It is in the A site. Okay, GTP gets hydrolyzed, and then that EF2 GDP leaves the ribosome. Okay, there are now 2 amino acids bound to tRNAs positioned for joining. One is in the A site, the one that just came in, and the other that had been there before is at the P site. Right, so that loads it. The next step in elongation is the forming of a bond between the 2 amino acids. So there are now 2 amino acids in the ribosome. Okay, N-formyl methionine, that first group, is transferred from its tRNA in the P site to the amino acid in the A site. So you get a transfer from the P site to the A site. Right? And this is catalyzed by the 23S ribosomal RNA. So in eukaryotes, that would be the 28S ribosomal RNA. The uncharged now, tRNA, is in the P site. So now you have an uncharged tRNA in the P site and a tRNA in the A site that has 2 amino acids attached to it. Right, so how this occurs, you get on the A site, the tRNA amino acid that just came in recently is going to have its amino group do a nucleophilic attack on the carboxyl group of that amino acid that's in the P site. So you get nucleophilic attack, and that's how you form that peptide bond. And by doing that, you're breaking the interaction between the 3' hydroxyl and this amino acid of the tRNA in the P site. Right, so you do that and you get this sort of a picture where you have 2 amino acids now and it's sort of sticking into the P site. Okay, the final step of elongation is what's known as translocation. So this is the moving of the ribosome relative to the messenger RNA. So the ribosome moves one codon towards the 3' end of the messenger RNA. And to do this, it uses energy from GTP hydrolysis. And the GTP is part of what's known as EFG translocase, also known as translocase. So elongation factor G is also called translocase. Okay, that leaves the A site open for a new amino acyl tRNA. Okay, so after translocation occurs, right, this whole thing shifts down, and the tRNA with 2 amino acids that was in the A site is now in the P site. Okay, and that leaves this A site open for the next one to come in. Okay, and then the empty tRNA that was just used, it just transferred its amino acid, is in the E site, standing for exit. So now it can leave. Okay, and this is all, this translocation is all catalyzed by that EFG cofactor. So EFG comes in, it has that GTP bound, it comes in to the A site, it hydrolyzes its GTP, which then provides a little bump to the ribosome to move down, which pushes that tRNA that was in the A site with the amino acid chain, pushes it into the P site. Right, and so now that EFG with GDP bound can dissociate, and it leaves that A site free. So now you can start that whole process over again. Okay, the final stage of translation is termination. And so in termination, you have what are known as RFs, or release factors, 1, 2, and 3. And the steps are to the hydrolysis of the terminal peptide tRNA bond, then the release of the free polypeptide and tRNA, and then finally the dissociation of the small and large subunits of the ribosome. Okay, termination is, it's not sort of random, right? It's signaled by a specific codon. And the 3 stop codons are shown here. Okay, when those are found in the A site, it's going to trigger the action of these termination factors, or release factors 1, 2, and 3. And these help to hydrolyze that peptide tRNA bond that's now in the P site, release the peptide and the tRNA from the ribosome, and then cause the subunits of the ribosome to dissociate, so that they can go and be recycled and initiation can start again. Okay. So what these release factors are, RF1 recognizes UAA and UAG specifically. RF2 recognizes UAA and UGA specifically. And then RF3 is used to release either RF1 or RF2 after the protein is hydrolyzed. Okay, so the first two recognize specific stop codons. The third one helps release the other release factors, okay, after the protein is released. You don't need to know the specific release factor 1 or 2. All you need to know that there is a release factor that comes in when that stop codon reaches the A site. The release factor comes into the A site. It causes cleaves the interaction, the bond between the tRNA and the peptide chain. It allows it to release. Then IF3 comes in. It involves GTP as well. And when it hydrolyzes GTP, it basically makes everything kind of explode into pieces, and they can be recycled and reused. The chemical reaction that is used to terminate translation, you have that peptide bond, that ester bond between the 3' hydroxyl on the terminal adenine base and the carboxyl group of the first amino acid there in the peptide chain. What happens is you get a simple hydrolysis. Okay, so water is your nucleophile here attacking that carboxyl group on the C-terminal end of the protein. Okay, and that releases it, and then your polypeptide then is given there. So pretty simple mechanism. Okay. So some features of protein synthesis. One, there's a very large energy cost to this, but that's important because most of our things and reactions that occur in the cell are catalyzed by proteins, by enzymes. So we need to pay this large cost to be able to control the environment within the cell. Okay. It can be rapid when accomplished on clusters of ribosomes. So clusters of ribosomes bound together, called a polysome, can really speed this process up. Okay. In bacteria, this is tightly coupled to transcription. So translation can begin before transcription is even finished, and that is shown on the title slide, or the first slide of the lecture. That was sort of the point of that, is that you can have in a bacteria cell, you can have a gene that's being transcribed into messenger RNA, and you can have ribosomes on that messenger RNA as it's being transcribed. Okay? Doesn't happen in eukaryotic cells because transcription is happening in the nucleus and translation occurs in the cytoplasm. So they're spatially separated. Okay. And so here's just a figure showing how this transcription and translation is coupled in bacteria. And here's actually what are referred to as polysomes, with you have multiple ribosomes attached. So you can get, in this example here, what we have 1, 2, 3, 4, 5 ribosomes that are on the same piece of messenger RNA. So you can get many copies of that protein from one messenger RNA, and it's even, occurring when this messenger RNA is being transcribed. Okay. Not surprisingly, many antibiotics target protein synthesis in bacteria. Things like tetracyclines, they block the A site on that bacterial ribosome. Chloramphenicol is a common antibiotic, at least one that's commonly used in the laboratory. These block the peptide transfer on the ribosome. Chloramphenicol also inhibits the mitochondrial and chloroplast ribosomes as well as bacteria. So it's something that is, not really used medicinally because of that. It's used in the laboratory when you need to select certain bacteria strains that are antibiotic resistance, for instance, ones that have a certain plasmid in them. Okay, and then also streptomycin. And this causes the code to be misread, which inhibits initiation at high concentrations. And their structures are shown here. Their structures aren't really, you don't need to know them for this class. But just know that this protein synthesis is a target for antibiotics. Right. Puromycin, it has a similar structure to the 3' end of the amino acyl tRNA. So it binds in the A site of ribosomes and forms a bond with a growing peptide, but it can't participate in translocation and dissociation. So that's another way in which you can inhibit protein synthesis in bacteria cells and use it as an antibiotic. Right. So the final stage, which is not really have anything to do with translation, but it's what occurs after the protein is translated. Stage 5 would be protein folding and post-translational modifications. So we'll go kind of quickly through these. Really not going to focus a whole lot on these in this class, but they are important, especially in eukaryotic cells. Right. So protein folding, that occurs, it's thought to occur as sort of a potential energy well, where you have an unfolded state would be up here, that has sort of high entropy. And then you sort of sample different local structures until you kind of come to a lower potential energy. Okay. Why do proteins fold the way they do? Well, they fold because of certain interactions, and those are charge-charge interactions, hydrogen bond interactions, hydrophobic force, and disulfide bonds. So interactions between side chains of amino acids in your protein chain. Right. There are certain N-terminal and C-terminal modifications that are made to proteins, and certain specific amino acids in the middle of a protein can also be modified. Things like the hydroxyl group of serine, threonine, and tyrosine are commonly phosphorylated, and we've seen examples of those throughout this year. Carbohydrate side chains are also added, and when that happens, that's referred to as a glycoprotein. Certain amino acids can be methylated or isoprenylated, so they can have these extra carbon or hydrocarbon groups attached to them. Okay. There are also signal sequences that we won't talk about them specifically in this class, but they're usually in the N-terminal side of a protein, and they tell you, C-terminal side, and they'll tell you where the protein should be located at in a cell. And those are cleaved by specific pepsidases once they reach the correct location. Okay, some post-translational modifications. In the eukaryotic genome, I want to say the majority of the proteins are going to have some sort of post-translational modifications. I don't have a good number, though, like a percentage-wise. But it's very prevalent in eukaryotic cells to have modifications made to amino acids in a protein after it's been translated. Okay. And some of these modifications are required before that protein becomes fully active or before its final conformation is achieved. They include the enzymatic removal of that formal group from that first methionine, or the removal of the methionine altogether, or sometimes additional residues with it. Also, acetylation of the N-terminal residue also occurs many times. Okay. The removal of signal sequences or other regions, and then also attaching carbohydrates or things like phosphate groups. Okay. All right, so amino acids themselves, they can be modified with additional carboxylic acids or phosphate groups. Isoprenyl groups can be added, and these are isoprene derivatives. We've talked about isoprenes when we talked about in chapter 10 lipids. Okay, that's just a very common building block that some lipids are made out of. And what these groups are usually used to do is to anchor proteins in membranes. So when you have an isoprene group attached to a protein, it really helps it be anchored in a membrane. Okay. Certain prosthetic groups can be added, things like iron-sulfur clusters, and then you can also form specific disulfide links. And if you have to form a complex between two separate proteins, disulfide links between two separate proteins can help form an active complex. Okay. By far, one of the biggest post-translational modifications are the addition of phosphate groups. And again, those happen onto amino acids that have hydroxyl in their side chain. So things like serine, threonine, and tyrosine can have phosphate groups added to them. What that does is it adds, obviously phosphate is negatively charged, so it adds a negative charge to it, which can alter the protein structure. These phosphates tend to come from ATP, and then special enzymes called kinases, which we've talked about most specifically in cell signaling, chapter 12, add those phosphate groups to them. Okay. These phosphate groups can be added to activate the protein or to deactivate the protein, so both. And the most recent example we saw of this was that C-terminal domain of RNA polymerase II in eukaryotic cells. That C-terminal domain is heavily phosphorylated to initiate transcription. Okay. All right, you can also add things like carboxylic acid groups. So in this case, we have a glutamate amino acid that has an extra carboxylic acid added to it. Sort of similar to adding a phosphate in that you're adding this extra negative charge to it. Right, some more modified amino acids are shown here. Methyl groups, another very common way amino acids are modified. What's happening here is that you're changing, sometimes you change a charge. You can also change, more importantly, the hydrophobicity by adding methyl groups. So these can also alter structures. Right, and then when we add longer groups, like the isoprene-derived groups, those can be added via this sort of mechanism, and they're added to like cysteine residues. So if you had a phosphorylated hydrophobic isoprene group, right, you can get a nucleophilic attack from your cysteine side chain, right? The pyrophosphate is removed, and now you have this longer isoprene that's hydrophobic. And in this case, it's our RAS protein that we talked about in the insulin pathway in chapter 12. Now it has this isoprene group on it, and it can be targeted and held into the membrane. All right, so that ends our discussion of chapter 27. I chose not to really cover any specific material in that third section. I think we've kind of covered enough material in this class. So hopefully this helps and makes sense. And the last 3 or 4 lectures isn't too much information, but hopefully it's enough that you kind of get an idea of this general concept of the central dogma and understanding how cells really work. And I know that you've probably seen some of this material in other classes, but hopefully this just gives you a little bit more chemical knowledge into these particular processes that are very important. If you do go on to grad school or medical school, you probably will be seeing these again, and you'll probably be seeing them in more detail, right? So just keep that in mind. All right, thank you for a great semester, and good luck on the final.